In this video, we will discuss about the efficiency of NanoCas editing in the non-human primates of muscle tissue. So what they will do is they will target this muscle tissue using the NanoCas editing, using the NanoCas uh, CRISPR gene with the targeted guide RNA in a AAV9-4A vectors, which is optimized for muscle muscle tissue deliveries. So how they start their study is that first they have to find out the guide RNAs that gives them uh, efficient editing at the muscle tissues. So what I did is, and for these studies they use the dystrophia gene, and this dystrophins is usually occurs in the muscle tissues that cause weakening of the muscle cells. So they wanted to target this dystrophin gene in the muscle tissues using the engineered NanoCas engineered system. So first they wanted to find out the guide RNAs that will give them maximum editing efficiencies or this disruptions. And what they are able to see is that they are able to find out two guide RNAs that gives them maximum efficiencies, and they named them as guide RNA 1 and guide RNA 2. And thereafter they prepared this guide RNA with this NanoCas9 along with this promoter CK80, which is expressed in the muscle tissues, and this WPRE and poly-A to stabilize these RNAs that are formed from this uh, transcriptions and translation of these vectors. And on the other end, for a control, they use this SA-Cas9 along with this CK80 promoter and the guide RNAs with this poly-A U6. And the size of this SA-Cas9 vectors is around 4.7, whereas NanoCas is 3.8. And then they use these vectors. So this is the size of the uh, this SA-Cas9 along with the guide RNAs sequence, and they then package them in this AAV9-4A vectors, and then do the studies on the NHP, that is cynomolgus maquus, and also on this mice. So before that they did their experiment also on this human myotubes, and there they see that positive results, that is results for this NanoCas9 is high compared to the this SA-Cas9, but that results are not shown in this figure. So here the results for this, first the mouse models are shown to target at this muscle tissues, and they are able to see these chromosomal editings are higher for this guide RNA 1 and guide RNA 2 to a range of around 30% for this quadriceps, and for calf the editing rates is around 25 to 30% also, this 25%, and for the hard tissues the range is around 12%, and for the guide the ranges varies from 10% to 20%, but they are not able to do this editings or compare it with the control of SA-Cas9 because of the problems, because the PAM sequence of this SA-Cas9 does not align with this single nucleotide polymorphisms at the site of these guide RNAs. That's why this SA-Cas9 editings cannot be seen in this mouse models, and but they can see the editings in this engineered NanoCas9, which D is 220 is mutated with R. Thereafter they check their editing efficiencies in these quadriceps also, and in the quadriceps they are in the non-human primates quadriceps they are able to see that with time the chromosomal editings are increases, that is from 10% to it rises to the 30%, whereas with this SA-Cas9 the editing rate does not increase and it lies within the 3% range. So this indicates that NanoCas9 has a better editing results compared to the SA-Cas9. Thereafter, they check this NHP editings at the cardiac tissues after 12 weeks, and here also they are able to see that editings with this NanoCas9 engineered gene is high compared to the SA-Cas9 gene for almost all the cases. All the cases. For example, this quadrichips is rates is around 30%, whereas in the SA-Cas9 it's around 2 to 3%. And similar trends are follows even for this heart rate. The rate of editings is around 15%, whereas in the SA-Cas9 it is around uh, 12%. So this indicates that the engineered NanoCas9 based NanoCas9 based delivery through this AAV9 4A vectors uh, has a better efficiency, a better editing efficiency than the non-human, than the SA-Cas9, and even the 10% increase in this dystrophins 10% editings in these dystrophin genes would help the patients uh, significantly or therapeutically. The 10% editings might give therapeutically significant relief to the patients, as it might helps in the recovery of these dystrophins edited genes. This is what has been clinically find out. And in the panel lab they have shown the different codings, coding length, coding domain, this coding sequence length, if they wanted to use different fusion proteins. If no fusion protein is used, its length is around 1300, whereas with AB fusion protein its length slightly increases to around 1900, and so on. It increases, it shows the actual coding strength, coding sequence length uh, with some of the popular fusion proteins. And all these are compatible with the AAV vectors. AAV vectors can hold up to 5 kilobase, 5 kilobase pair long DNA sequence. So I hope you understood how they validate these NanoCas editings in the non-human primates of the muscle tissues, and their results are positive and gives the higher efficiency compared to the uh, traditionally available CRISPR-Cas9 system, that is SA-Cas9. If you want to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.